Welcome to Deep Matters. Today we're talking to Geoffrey Dubourg-Felonneau about protein discovery and machine learning. Geoffrey works at a company called Shiru, and they're compiling a large data set of naturally occurring proteins, the kinds of proteins that the body can absorb and digest relatively easily, and they can then therefore serve as useful substitutes for foods, like meat substitutes, for example. But in order to do that, they need to have their own wet lab, and that wet lab is helping them to determine the kinds of properties and characteristics and functions of various proteins. And then they're using machine learning to model that space of protein sequence to function. We're going to go into detail about how similar machine learning models can be built, what the data looks like, what the architecture looks like. We're going to follow a paper very closely that they had just released, not detailing exactly what they're doing, but detailing something quite similar and relevant. We're also going to be talking about that loop that they have between the wet lab and their machine learning. The wet lab is producing the kind of data that is then going into their machine learning. The machine learning models are then able to suggest the next experiments that make sense to run in the wet lab. This was a very fun conversation. Geoffrey, thank you very much for coming to the show. Let's dive in. I'll tell you how I came across you guys. So basically, my girlfriend and I, we were dating for a few years and we broke up a few months ago, right? And then I decided, okay, I need to be in better shape now, right? And I got to eat healthy. So I started going to the gym and I started eating healthier. And I started, I was at Trader Joe's the other day and I bought like non-fat cottage cheese, right? And I just brought it, next to me to my computer, and I was eating it. And I noticed it had this kind of like watery, it had like a watery film to it that I normally don't see in just like regular kind of like whole milk cottage cheese. And I was thinking, well, now I have ChatGPT next to me. Why don't I just ask it, what the hell is going on? Right? Like, why is there, and I've noticed it in a couple of non-fat cottage. It wasn't just like this one, right? So I started, anyway, I was down a rabbit hole with ChatGPT, and it was telling me about adding stabilizers and thickeners and all these different things. And I ended up coming across this, and I asked it, well, what are the typical kind of like stabilizers that they add to non-fat cottage cheese? And they said one of the things, I think it's called carrageenan. And I was like, okay, that's interesting. Where is that produced? And it said in the Philippines and in Indonesia, and it comes out of I think like seaweed. Yeah, it comes out of seaweed. And then I was like, and then I asked ChatGPT, who are all the companies that are manufacturing this sort of thing? And they gave me one name, and I think it was like CP Kelco.
CP Kelco, yeah.
CP Kelco. And then I looked up, I was like, okay, I wonder if they're using machine learning for this. So then I looked up, I was like, CP Kelco plus machine learning. And then I saw that they just partnered up with a startup that's doing some machine learning stuff. And that's you guys. So that's how I ended up with you. So
yeah, so we're at the end of a long trail of
this
search. And cool, yeah, that's a fun story.
How did you end up there? What's been your background?
Yeah, so my background is in 2015 I graduated so from master degrees in engineering for computer science and engineering, and then I moved to San Francisco and I started to work for a startup, very small startup doing AI for music, basically how to use generative AI. So back in 2015 was not as powerful as now, but yet we had good ways to do generative AI for music. I worked there for 2 years, and then I kind of randomly or by accident, followed my wife who were doing her master's degree in Cambridge in the UK, and I found this great company called CCG, Cambridge Cancer Genomics, and they really introduced me to the world of proteomics and multiomics, and I used my background in how to use machine learning that deals with sequences in music to like DNA sequences or multiomic sequences. And I worked there for 2-3 years, and then I moved back to the Bay Area in 2020, and and I found Shiru which is doing a quite exciting mission. And again, it's a lot of creativity, a lot of research, engineering, and one of the rare company where we have, a dry lab doing all the engineering and all the research and all the building hypotheses, etc. But also a wet lab where we actually test things in vivo, and we actually generate data that we will use for our algorithms. So having everything in the same place is super exciting.
Yeah. So interesting. So your background, formally your background's been in the machine learning, the data science, and then when you then went to the UK, how much of the biology did you need to pick up, and how did you do that?
Yeah, so I had like a tiny little background of biology in a kind of high school biology. I was kind of took the branch I specialized in biology, but really not enough.
Yeah.
And really I relied on the patience of at the time my CTO and other team members who were super good at basically telling me everything I needed to know and also guiding me to the resources. And it's sure that I had this kind of entry into the biology world through the lens of machine learning. So I usually learn about biology concepts through machine learning paper usually. So I'm like, oh, so there is this question and that's how you solve it, rather than studying like the base of biology and then trying to solve it like this.
Yeah. Okay, cool. So you mentioned a couple of words that I want us to sort of unpack. Proteomics, multiomics. What are these things for people who are approaching as you were from machine learning? What are these things?
No, yeah, I think it's just a fancy word to say about all the type of data that relates to, so proteomics, all the type of data relates to proteins, and the type, also the methods you will apply on this data. Multiomics is the methods that Ensemble, or that at least can handle proteomic data, so protein data, genomic data, so DNA, and transcriptomic data, so RNA data. All those are sequences, but they are kind of related, but means different things. And we have, there are models, or more like, I would say, pipelines that can handle all this data to extract some meaningful insights. So multiomics will be like a combination of at least two of those things. And proteomics is really what we do at Shiru, focusing on the proteins.
Okay, so what do you guys do at Shiro?
Yeah, it's a good question. So we are finding alternative proteins for the food industry. As you just mentioned, there are when we want to go for a food product that doesn't have any animal proteins, we have some caveats, because people in the industry use animal protein for a reason, because they have some functions. The proteins that come from animal products have some useful function that we kind of discovered over the centuries. Now what we're trying to do is to find those proteins that are not from animal sources, but carry the same function, or at least very similar functions. And as things like stabilizer, or holding, making a structured fat, for example, is our last product called oleogel. So if you want to, something that make a kind of a matrix of fat that is holding itself, there are animal protein that can do that, and we found non-animal protein that do similar things.
So when you say function, it's a technical term.
Right, function is a technical term, and also it's quite interesting, because when you talk about protein function in the literature, you will very often find function as in vivo function, as in like this, what is the function of this protein in the human body, for example. But that's not what we're interested in. We're interested in the food function. So how does this protein behave once it's in a food matrix at a given temperature, at a given whatever food environment? And there is no data set for that. I mean, public at least. So we're building the data set, and we're learning, and we're using our limited knowledge, because experiments are expensive and building a data set is expensive. So we're really using machine learning to extend the knowledge of these data points to propagate basically the labels to the world of proteins.
Okay, I want to understand in a little bit more detail why proteins and not, let's say, like any other kind of substance, ingredient, macromolecule. Like, what's so unique about proteins?
Right.
Yeah.
That's a very good question that I actually rarely asked. It's really at the core of the company, and we're so doing protein and not small molecules. One of the reasons I believe protein is very very interesting. First, it's hard to work with proteins, so that's why not a lot of companies do that. So precision fermentation, so making just one protein of interest instead of filtering through a junk of protein and just extracting what you want, is a new field. So it's not that easy. But why would you be interested in proteins? Because I would say there are basically when you ingest a protein, your body is trained to break down proteins and knows how amino acid works, and there will be after the stomach there will be like very little of this protein. This protein will be broken down to amino acids. That is a kind of non-component to the human body. Whereas if you start doing some kind of new small molecules that are maybe easier to predict in their behavior, their impact on health, I think, is harder to anticipate.
That's very interesting.
That's my understanding, and that's why drug discovery would focus on small molecules, but have to go through a lot of testing.
Yeah. And you guys you're gonna have to deal with safety issues as well, but this
right.
At least it's a little bit less.
Yeah, a little bit less. And when you add on top of this that we are currently only focusing on wild type proteins, so proteins that exist in nature. We're not engineering proteins. We're using protein that exists.
Is it then fair to say that most of the ones that exist already out in nature tend to be? Are there any toxic ones, or that we have to worry about?
For sure, yeah, no, there are toxic proteins. There are allergenic proteins as well. There's a lot of protein you might not want to ingest. We do have models that are trained to detect that, as well as a combination of labeled data set. I was saying that the food function is not necessarily labeled publicly. Toxicity and allergenicity are things that are better labeled publicly.
Yeah, so I'm gonna want to ask you about that in a little bit. So so walk me through it. Let's say there's a company and they want to produce they want some plant-based protein to replace their animal protein for some, like, what should I have in mind? Is it like a vegan hamburger?
Yeah, we're working on a bunch of different functions. I'm not sure how much I can disclose which function we're working on. There are things that are public, so the structured fat is definitely public. But yeah, so your industrial, you make some fluid, you have a function in mind. This function is mainly carried by either a small molecule that is not clean label or a protein that is from animal sources, and you want to replace those products.
And is the reason they want to replace it is it just because is it cheaper? Is it that people are becoming vegetarians? Like, what is driving this?
That actually, I wish there was a good answer, or at least one answer, but it's probably a combination of factors. That yeah, first the cost is always one of the main factors. So the cheaper, the better, that's for sure. But on top of this, yeah, there are like environmental concerns for proteins that are animal based. You can also feed the vegan market if you can make the same product without any animal products. So yeah, it's a combination of factors, and ideally, we would love to get to the same or better price point, same amount of deliciousness, and without having a bad impact, negative impact on the environment.
Got it. Okay, so company X comes to you and says, "This is our current set of ingredients. This is, we want this to be consumed at this temperature. We want it to have this sort of texture. These are sort of all the requirements." And then they tell you, "Go and fetch for us or find for us or discover the kind of protein that can best substitute one or multiple of these animal-based proteins," something like
Correct.
that?
Yeah. That's pretty much how it
Is
works.
that a service that you provide, or is that a product that you've built that they can now use?
It's more of a service because there is a lot of education. There is a lot of first coming up with the specification is already a lot of work. Getting to know exactly what you want to replace, and yeah. So once you have that, you build this specification. That's we use our internal protein search engine coupled with a different machine learning pipeline to come up with a list of candidates that then we test in-house to validate that they indeed carry the function of interest. And not only they carry the function of interest, but they're also manufacturable. So you can make them at a large scale because we tend to forget, but precision fermentation is not magic. We're still working with organisms that cannot do whatever they want, and you need to understand them to make them produce your protein or a protein. And it's not, they would not produce any sequence. They would not produce any proteins. So it's a combination of what will work the best, so as in function wise, but also what is going to be a good manufacturable candidate.
Okay, got it. So I want to know much more about that machine learning pipeline and process. But if we just like black box this for a second, then what ends up happening is you come up with a bunch of candidate proteins, and then you're going to check each one of them. Now, each of these candidate proteins, you already know how to source it, right? Because they've all come from the natural world. Is that a relatively easy bridge to cross? Like, given a protein, do you know where it comes from? Or has that information sort of
it's
been
all
detached?
it's one of the public database fields. You know the exact organisms it was sequenced from.
So then it's okay, this protein comes from this type of beet, let's say.
Right.
Then you have to say, okay, but can I actually do this? Can I manufacture this at an enormous enough scale? So then you have to go through a bunch of like experimentation on your own. This is kind of like where the wet lab comes in, and just try to produce a lot of beets and make sure that they consistently give you those kinds of proteins. Is that the idea?
So the thing with precision fermentation is that we're using organisms. So it could be yeast, could be other things, to make this organism really secrete or produce the protein, only the protein you want. So it's not we were getting the whole organism that we have to put in a blender and extract the protein. We really ask this the same way you would make beer and you would have yeast making ethanol. You could also have yeast or other organism making a protein of interest, and that's what precision fermentation is.
That's what precision fermentation is. I have no idea what that is, but that's very cool. Okay, I'm looking at it. So with fermentation, so I guess it's kind of that's what happens with fermentation of just like pickles too, right? Like it's the certain type of bacteria or something that is pulling out some of the nutrients, right? And I don't know what else.
Right.
Like, what is the magic of fermentation? What happens then?
Well, it's probably way better to ask a fermentation expert because there's so much happening in fermentation, and also I recently learned that fermentation can you can ferment many different things. So as I was saying, you can ferment ethanol, you can ferment like a product, like a small molecule, but you can ferment also like make a protein because it's just basically you just ending up with like organisms living and making sure that the byproduct is something you're interested in. So they end up they just want to leave, but in the process of doing so they secrete or they make something, and that's what you want. And
that's interesting.
It can be so many different things. So I think precision fermentation has a lot of has a bright future because now there are more and more techniques to really program those organisms so that they really make what you want. It's not as easy as it sounds, but I think it's very interesting. And
that part of the process that's not where the machine learning comes in yet, right? Although I imagine there might be like other kind of that's not where like your protein discovery platform is targeting, right? It's not about the fermentation.
It's not about, so we're trying to incorporate machine learning at every stage of wherever there is data, wherever there's a problem like a task to optimize, we try to apply machine learning. So it depends the approach or the angle we're talking about, but ideally we're trying to solve all our problems with machine learning. Now there are data consistency issues or data availability or power of data issues, but we definitely have the intent to solve everything we can solve with machine learning.
So I want to dive a little bit deeper into how you guys are using some of that now. So a client comes to you, and let's imagine that you've worked with them, and now you've been able to kind of distill all of their requirements into some kind of more formal setting, right? So you say, okay, these have to be the temperatures, this has to be the texture, and I imagine each one of these things is sort of tethered to something quite quantitative, right? Or measurable. And then you go through your own, you have a database, you're trying to identify candidate proteins. What does the machine learning look like here? What are we talking about?
So there's a lot I cannot talk about, some I can talk about, which is the paper essence.
So this paper is like, this is a contribution that you guys have made to the community, and I don't know whether that's similar to how you guys are doing it in-house, but we'll be able to talk about that as a process.
I think it has some good amount of overlap, enough to kind of get a good understanding of how things will work in a production level. And in that case, we're talking about regression or classification of proteins or label propagation. So we have in-house experiments that label proteins, and some customer come and they want a protein that has the combination of label, right? We do not have it, or if we already have label, it's great. Unlikely, but great. If their specification falls into a range of something that we don't have, then we have to look for the predicted values. And That's where we use models from a protein sequence predict some features of interest. I mean, some label of interest.
In the paper, you give the example of localization.
Exactly, yeah. In the paper, we focus on a public data set called, that is used in the paper DeepLock2. So it's a public data set that, yeah,
I think I have this.
that focuses on localization, so cellular localization. So the protein, once it has expressed, once it exists within the cell, travels within the cell or outside of the cell, and where it ends up is called the subcellular localization. And knowing where a protein is going to end up can be very useful because, again, for production, you might not want the protein to end up in some part of the cells for scalability. Knowing where it ends up in the cell also allows you to infer some of its characteristics. So it's a quite useful task to be able to do, and there is a public data set, so it's very
that helps.
nice to write.
Oh, I see. So okay, so you're using where it ends up, sort of like the destination within the cell, as some marker or some attribute of the protein. And that attribute can then tell you, or perhaps correlate with other things that you might actually care about from like a business perspective.
Right.
So customers care about X. You don't yet have information about X, but you have information about Y, and Y here being like where exactly it ends up in the cell. But there is some type of relationship between X and Y.
Exactly. Yeah. It's unless a customer wants proteins that end up at a specific location in the cell, which is unlikely. It's more like being in. For example, I think we can take the example of being predicted to cross the cell membrane, so to be an extracellular protein can have some significant advantage for being produced in a large scale production. So if you can predict that the likelihood of a protein or given sequence to actually be labeled extracellular allows you to really sort your candidates in a more informed way.
Yeah, that's very cool. Okay, so just while we're still on this topic, subcellular localization. So where can it end up in the cell? What are the possible destinations?
Yeah, so I think there's 8 labels, so there's probably more in the paper, but there's more in reality. But if you look at in the paper, we end up with things like I think we made a result table, which are the cytoplasm, the nucleus, the cell membrane, the mitochondrion, and those are at least the one we're interested in. Those are just positioned in the cell. Again, we use this as more like a downstream benchmark task to show that this method could be applied to any labels, and in reality, you would not necessarily have those labels. You could have like other labels, but the method would stay the same. So this paper, which is nice about this paper, is that you could totally change the data set. We can expect the result to be similar.
So the data here, okay, so the idea here is you already started with some like publicly available data set, right? Somebody's labeled this, right? This was supervised. So how many proteins are we talking about here?
I don't have it on top of my mind.
But it's like
I think
hundreds of thousands, tens of thousands.
Yeah. So If we talk about proteins available for which we have a sequence, we're talking about hundreds of millions.
Yeah.
Protein in our data set or in our database, which are plant-based, they are like 30 to 40 millions that we have. And in this data set, if you look at the,
here
yeah, we have
24,000.
24K
Got it.
that are labeled. There are more that could be labeled with those type of labels. But every time you go through kind of building a machine learning data set from a biological data set, you have to incorporate a lot of domain knowledge and making sure that your sampling is okay, that you're not over-representing some part of the space, etc.
Yeah.
So having something that's already built saves you a lot of time.
How do you have a sense for how this is built? So like 24,000, like, do they actually have to look protein by protein and just like see where it ends up in a cell? I mean, that just sounds like an enormous amount of work.
Yes. So it's not the people who I mean, I think that's the people who compiled the data set who did that work of like looking into the cell. But if you look at basically the world of proteins of known sequences, there are two types of labels. There are labels. So, for example, if you take the data set called UniProt or the database called UniProt, a small part of it, I think 1%, is called SwissProt, which is all the proteins for which we have a high quality labeling. So where indeed there are people, who spend a lot of time researching and measuring things and writing papers about their findings, and we end up, I think so. Yeah, exactly. Yeah. So UniProt, you'll have so that's going to be the tool that a lot of proteomics people will spend some time on. But basically, you have I think if you go down a little bit, you can see the numbers. SwissProt up a little bit. Here, your proteins, UniProt knowledge. You have unreviewed, and you have 249 million.
Yeah,
and SwissProt, which have been reviewed, which is more than 500,000. So that's and what is not reviewed is often predicted by some machine learning approach and some more canonical bioinformatic methods as well.
So, okay, so walk us through the challenge here of, let's say, even just like solving this problem. So you have a sequence, a sequence of amino acids. That's how do you even represent the protein?
Correct.
Okay, so a protein is just a bunch of amino acids. And they're organized in some kind of bizarre shape or form or something like that. But really, it's just a list.
Yeah, it's exactly. It's a list. It's a sequence. The way it's given to the computer. Now, what gives a function to a protein is actually a combination of mainly its overall 3D structure, and which part moves in the structure, which parts are rigid, and two, the biochemical characteristics of each amino acid, plus where they are. So, for example, if you have some amino acids that are hydrophobic, right? They repel water. If they are outside, if with the final 3D structure they are outside, the whole protein is going to be hydrophobic. But if somehow they're embedded and hydrophilic protein are outside, then the protein is going to be hydrophilic. So it's a combination of a lot of factors that end up to what the function is going to be. And
when you look at a data set, like at a database like this, if we were to download this, each row would be what? Just like a sequence of amino acids, and how there are 20 amino acids? right? How many amino acids?
There are, yeah, 20 amino acids.
So it would just be acid 1, acid 5, then acid 9, then acid 1 again, acid, right? It would
so
just...
the vocabulary is 20. Yeah, the vocabulary is 20 amino acids, and the sequence length can be anywhere between 20 and 2000 or even more.
Yeah.
So you have it's like every protein is a sentence basically, and you have a corpus of text, and you want to classify the sentences. Let's say, yeah.
Now, in this case, if the only thing we had is a is just, let's say, like the alphabet, right? So we just like that sentence. That still doesn't tell us anything about the shape of it, right?
Exactly.
So, okay, so let's imagine you have that sequence, and what you're trying to classify for this model is the destination, right? The localized kind of where it's going to be. Okay, so before there was machine learning applied to this, are there some trivial ways by which you could just sort of by eye look at the sequence and say, oh yeah, this is going to end up here and there?
Yeah. So given a data set, what people would do mainly is look at sequence identity. So given some sequences that you already know go in some places, if you get a new sequence, what you could do is running this new sequence against all known sequence and see how similar it is. And if it's very similar to something we know end up in the nucleus, for example, you could you can make the hypothesis that it would also end up in the nucleus. This approach has a lot of caveats where machine learning and specifically language models can come into play here. But that's, I think, again, I'm not a good canonical bioinformatician, but that's definitely what people used to do.
Got it. Okay, so now you have that sequence, and so what is the architecture that you're going to follow to solve this problem?
Right. So what we've done, there are a couple of available architectures, and what we do in this paper is actually combining two architectures and making an ensembling of those two already powerful architectures, and we show that it gets even more powerful. So the first one is going to be based on language models. So the top part, where you give a sequence and you feed that to a language model, and it's really BERT for proteins. A BERT trained on protein where each amino acid would be a word, and the whole protein would be a sentence, and the BERT has just learned how to reconstruct the sentence or the protein with the missing amino acids. In BERT, you also have the next sentence prediction. They don't do this. In general, people who train proteins, BERT for proteins don't do this because you don't have a protein that follows another protein.
Yeah.
I think there might be ways of using next sentence prediction for other tasks that involves proteins, but overall in the community, we'll see people just focusing on the masked amino acid task.
So the way that it was trained, it'd be like, let's say you have 20,000 different amino acids. Oh no, sorry, 20,000 different, amino acids, right? It could
amino acids, yeah. I think we can, in average, the length is in the 500.
So let's say you have 500 amino acids. It will mask the first one and then try to predict it on the basis of the other 499, and then just kind of sequentially mask more and more in the same way that you would do it for language. Okay. And these are all just like transformer based. They're very similar to, yeah.
Correct. Yeah, it's literally a BERT.
Yeah. Okay. And who produces? These are all open source?
These are almost. So there are some open source, some less open source. I can cite a few. So the one we're mainly using in the paper is ESM, that is produced by Facebook. I think it's evolutionary scale something. Yeah.
Scale modeling, yeah, that one.
Yeah, modeling. So ESM is very nice because it gives you a lot of different models that are quite updated quite often. So that's yeah, and basically if you follow the code here, you can see that you get from a given sequence, you can end up with a representation. So so here's for structure prediction. They went quite far, and it was really nice to follow this GitHub in for as time went, because yeah, now it's like a lot of layers on top of previous knowledge. And
you're using the embeddings though.
Yeah, yeah. For example, yeah, things you can do like this. Yeah, so embeddings is one of the things we're using for representations at Shiru, and in this paper, yeah, that's the one we're using.
Okay, cool. So basically, you feed in the sequence, and you get a bunch of representation. You get this sort of like embedding. So this is the language model representation, and okay, I cut you off. So this was the first approach. First approach is to just go the language model route.
Right. And here you could train, ahead of the model or a couple of last layers. So you can freeze this whole transformer encoder layers and just fine tune basically the last bits for a downstream task, like here the subcellular localization. And frankly, you would have already a very good result. This worked very well. Yeah.
Okay, so that's sort of so that's one approach. You guys then combine this in an ensemble. So what's the other one? Yeah.
Right. So the idea was to when we thought about this paper, AlphaFold just came out, and we were like, okay, for sure. So we're giving the sequence to the model and giving the information about the structure. For sure, will make it better, right? Because as we said, I mean, depending on the downstream task, but here we're benchmarking for subcellular localization. We believe that giving not only the sequence, but the overall, I mean, the information about the structure will really help them all, making any type of inferences. So what we did is we used the output of AlphaFold, which are PDB files.
What does it look like? What is this PDB file?
So PDB file it's canonically what is used for representing 3D structure. Yeah, 3D. So it's a little bit confusing because PDB is the name of the database and also the name of the file. But yeah, it's a set of coordinates where you would have each atom in the molecule. Each atom of each amino acid, and you see C, it means carbon, but there is also Ca, which is the carbon alpha, which is kind of the center of the amino acid. And so AlphaFold really gives you every single atom. But if you want to focus on kind of the overall shape, you can just take the carbon alphas, the chain of carbon alphas, and it will tell you overall, where each amino acid end up.
Oh, that's cool. Almost like the center of mass.
Yeah. It's where it's what makes the difference between the backbone and the side chain. And the side chain kind of it's another layer of complexity. Is one of the reasons it was so hard to fold proteins in silico. Because so the whole backbone can fold, but also the side chain can move around and impact the folding.
So okay, so you feed it into AlphaFold, and basically what you get is something like this. And now you're going to convert this into some form of graph.
Correct. Yes. So from here, we decided to build a graph where each node is basically the carbon alpha I was talking about. So basically representing the amino acid, and there is an edge if two carbon alpha are close enough, and that will give you a graph of overall structure of the protein.
Okay, so a couple of questions. Why, if they're close enough, put an edge as opposed to always add an edge and add a weight that corresponds to the distance?
Yeah, that's a good question. So, well, if you want to use a graph-based approach, you probably don't want your graph to be fully connected for either computational reasons and also learning patterns. If you do some pooling, for example, so some graph pooling, you want the graph to have a topology. You don't want everything to be connected and be solely relying on the weight of the edge.
Weights. Sure.
I mean, technically you could, but then you might just want your program to be learning at, which weight you kind of discard an edge to extract the topology. So
it sounds like a heavier learning task. You might need much more data and hyperparameter optimization.
Right. It's one of the things where here you can pick a threshold that is informed by experts, where we know that two amino acids are actually like chemically connected if they are below a given threshold. So we can use those thresholds and this knowledge.
Yeah, that's cool. And then, is this also a similar reason for why you decided to only focus on the carbon alpha as opposed to like just the entire base? Okay.
Yeah, basically baby steps. We try the first thing, we see if it works, and then if it works well, we kind of move forward. Making or training a graph that represents every single atom might come with some other computational challenges for sure.
Yeah. Okay. So you feed it all into this graph. And well, first you represent it as a graph, and then you feed it into some graph neural network, and you get some form of these are like similar as like embeddings or something.
Right. Yeah. Then you can extract the representation as well.
Got it. Okay. And I think I see here. I mean, this maybe for people who are just tuning in for audio. The idea is that you then sort of concatenate both representations. Yeah,
it's just a fancy way of with trial and error, we ended up with this ensembling architecture. You could have just focused on the mean or train, concatenate and train the final layer. And with couple of iteration, we ended up finding this kind of random architecture that worked the best.
Very cool. Okay, so nice. Now this ensemble is the thing that wins. This is way better than each one of these alone, right? How good are these models in the first place?
Right. So if you go on the result table, you can see that.
Let's see.
And so long story short, you could focus here on the F1 score. So here on the left results. And I think the big kind of conclusion of this paper, or what this allowed to us to understand, is that overall language models are. I mean, the way it was trained, because also language model comes with a very unfair advantage. It has been trained on hundreds of millions of proteins, and our GNN has been trained just on this task. So obviously you have a kind of an unfair comparison here, but the GNN we've trained, if you use it alone, and so it's going to be the table on the top, the first row, you have a F1 score of 0.46. And if you do the language model alone, you have a F1 score of 55.
Yeah.
So already the language model is better for one of the reasons I said. And the second reason, maybe you can get into it, is this more philosophical. It's kind of it's known that language models actually learn about the structure. So that's something to keep in mind. And if you take the final row of this table, we see that with ensembling, we end up with a 0.6. So we beat both of them independently by ensembling them the way we did.
I'm very curious about, so this is similar to what you guys are implemented at Shiru, or at least it has some sort of overlap. But I imagine that you guys are doing many more things, because what you have to do is much more than just to localize within a cell, right?
Correct.
So you have some more like fancy thing going on. But one thing that I find very interesting, and I think you alluded to it right in the beginning, is that you guys also then have the wet lab component of it, right? Like, so you are able to then continually generate more and more data.
Right.
Can you is there something you can tell us about that process? Because that's the thing that feels quite distinct from what you would normally see in like a machine learning powered company, right? Like, how expensive is it? Not just money wise, but just kind of like effort wise. How long does it take to just get like a single protein in? Is this just a massive operation? Are there hundreds of people that have to do this, or is it just like one kind of chemist pushing along?
Yeah.
Like, what's the picture?
No, it's a very good question. And so I guess to give an idea, we are 40 people in the company. And I'd say more than half or to 3/4 of the company is really focused on that activity, the wet lab. So given a sequence, generate first the protein, and two, the capture interesting labels and interesting characteristics about this protein. This is quite hard, particularly for an early startup. But I think we're doing very well, because we're already able to generate hundreds of proteins at a time. So not just one or two proteins. We're doing batches where we're talking hundreds multiple times, and we have multiple thousands of data points, depending how you count, but like thousands to tens of thousands of data points. So this is quite intense, because there's the kind of the whole pipeline to get from the sequence to the generation of the data of interest is long and involves a lot of different type of experts. We're talking strain engineering, talking about fermentation, we're talking about high throughput, people programming robots
how automated is this process? Because like, well, you certainly need experts in the room.
Correct. Yeah, we have a fair amount of automation with experts in basically robotics, building and coming up with the building the pipeline to make all the operations you need in a very consistent and very high throughput way. All this flow, you could do it with basically people using pipettes and looking through microscopes and pipetting things. Most of this flow for us is automated, which really allows us to go hundreds of protein at a time, if not soon thousands. So, and this really is mandatory to apply machine learning, because machine learning requires high quality data, if not like a lot of more data, so that you can make up for the noise. You can buy noise with
volume,
data size, with volume. Since we're not talking about millions, we need high quality data, so consistent, repeatable data, where environmental variable conditions are measured and consistent is very important.
But even then, it sounds to me like an enormous task, because even if you're analyzing, let's say, a single protein in isolation, well, you're gonna have how does this protein react to all the other ingredients, right? And how does it react to the way in which it was processed, and how does it, like, you just have so many variables that, first of all, it feels like you can't possibly do this by hand, right? You're gonna need some kind of automated way of doing it. What is your, if we could just, like, let's say, like, fast forward 5 years, 10 years, 15 years, what are you imagining is sort of like the future of either like protein discovery or just like food ingredients, its relationship with data? Do you have some thoughts about this?
Yeah, I mean, obviously I'm very optimistic. That's why I'm working here. So I really think that we have all the tools. So we have data on millions of proteins. We have high throughput experiments for labeling what's missing for downstream tasks, and we have very powerful machine learning models. And what's also very exciting is that models that works well in some fields where you would apply machine learning are very transferable for this task, for this type of tasks as well. So with all those three together, I think you can easily, and we've shown that you can easily found this next set of proteins that will carry the task you want. Then for sure there will always be challenges down the road, and I think that's why it's exciting, because you need to tackle them. But I'm quite optimistic about the field.
I wish you guys a lot of success and a lot of luck. I've been a fan of the synthetic meats industry kind of burgeoning, and I think there's so much promise there. So thank you for all the work that you're doing. If people want to get in touch or learn more either about the company or about you, where should we send them?
On Shiru.com. Yeah,
Shiru.com.
That would be a good start. Then we try to go to machine learning conferences like New Rips every year. But Shiru.com will be the first stop.
Well, I'll make sure I'll add the link in the show note below. Geoffrey, thank you very much for joining us.
Thank you, Adam.